Hi, I am Ankur Gupta from Department of Chemical Sciences, Tata Institute of Fundamental Research, Mumbai. And today I welcome you all to my show Wonders of Nature. And in today's show we have our guest Brain, our brain.
Hello friends, I hope you all identify me. I live in your head and manages your whole body. My main job is to control and perform your various body functions, like I help you in thinking, breathing, moving your muscles, and I'm still awake when you're sleeping. Have you ever wondered how I perform all these body functions? Like when you get a complicated homework, you take help from your friends. In the same way, to do all these complicated jobs, I have a group of special friends known as neurons. I have millions of such neurons which communicate among themselves to pass my instructions to complete the job.
The talking of neurons to each other is known as neurotransmission. Now,
let's start with our demonstration. Suppose you wanted to lift a box. Your brain neuron will generate a secret message in the form of a nerve impulse. The special machines inside the neurons, which are known as snare proteins, carry small packets called synaptic vesicles. These packets are filled with small molecules known as neurotransmitters. These snare proteins release neurotransmitters from the vesicles, which fall on the magical net called membrane of the another neuron. The membrane contains traps called receptors, which recognize the released neurotransmitters. These receptors grab the neurotransmitters and get activated. As a result, a series of biological machines are activated and they help to decode the message from the first neuron. In this way, the muscles, which are very far from the brain, receive the message, and finally you can lift the box. This way of communication mainly happens through the receptors and is known as receptor-mediated neurotransmission.
this neurotransmission is the sole basis of many functions in our body. Any disruption in the process of neurotransmission causes many diseases associated with the malfunctioning of our special guest brain, such as mental illness, depression, etc. I cannot imagine my life if my brain stopped working. Can you? Do you remember watching any crime serial series? It is always very exciting to see how the detective uses each and every detail of the crime scene to solve the case. Similarly, to find reasons of brain malfunctioning, it is extremely important to understand each and every step involved in the process of neurotransmitter. Scientists all over the world have actually carried out millions of experiments to understand how this neurotransmission functions, and they have proposed a very well-known mechanism known as receptor-mediated neurotransmission.
In the neurotransmission, you may have noticed that only few neurotransmitters are grabbed by the receptors, while many neurotransmitters got stuck to the membrane.
Then why so many neurotransmitters are thrown into the membrane? Do these neurotransmitters love to bind to the membrane? If so, does this binding have anything to do with the process of the neurotransmission? With all such mysteries, I started my journey in the lab. We make these membranes artificially. So I decided to calculate the binding of these neurotransmitters with the membrane. I picked few neurotransmitters such as serotonin, dopamine, norepinephrine and epinephrine and calculate the membrane bound amount of these neurotransmitters. For that, I carried the simple experiment. So I made artificial membranes and added the neurotransmitters on top of that. After that, I separated the membrane bound neurotransmitters and measured its amount. I found that these neurotransmitters have very high binding affinity with the membrane. If you find that such a large amount of neurotransmitters are hitting the membrane and getting bound to it, then it is highly possible that they could change the membrane physical properties. For example, the membrane can get loosened. So I decided to measure that how the physical properties of the membrane change in the presence and absence of neurotransmitter. For that, I picked the instrument known as atomic force microscope and calculate the membrane mechanical stiffness in the presence of these neurotransmitters. Let me first demonstrate you how this atomic force microscope is used to measure the mechanical stiffness of the membrane.
In the atomic force microscope, we have a tip like a needle with the nanometer dimension. With the help of the motor, we move the tip towards the membrane. We poke the tip into the membrane and measure the force required. As you move closer and closer to the membrane, the force increases, and at some point the tip breaks the membrane locally and we get a kink. The force corresponding to the kink is a measure of mechanical strength of the membrane. Simply, higher the force required to break the membrane, higher will be the membrane strength.
So, I found that these neurotransmitters can change the mechanical strength of the membrane. In addition to this, I also found that these neurotransmitters can change the mechanical strength of the synaptic vesicles in which they are filled. So, in today's show, we learned that in addition to this receptor-mediated neurotransmission, there is a role of membranes in the neurotransmission and which gives rise to the membrane-mediated neurotransmission. So, to treat the malfunctioning of the brain, scientists can also focus on the role of membranes to keep it happy and healthy. So, this is all for today's show, Wonders of Nature. Thank you very much. Bye-bye.